All right. All right. Hi, everyone. Welcome. Tonight's topic is, I think, an extremely exciting one. It's very relevant. It's very much in the now. We're talking about culture expanding autologous stem cells. So stem cells coming from you. We have the CEO for a company called Forever Labs today with us, Kevin Virgil. And Kevin's going to give us some information about their culture expansion lab that's being built in Florida right now, as well as the ability to bank your own cells. But before we get into that, we actually wanted to talk to Kevin about his experience as a patient. So he's also a patient of ours. Michigan Center of Regenerative Medicine treated his knees about a year ago. And if you wouldn't mind, Kevin, just give us a brief background on kind of how you developed knee problems, what your history was, what your trauma histories were, and then kind of take us up to speed where you're at now.
Yeah, no, thanks very much for having me tonight. And yeah, I guess I had I guess you'd call it a fortunate opportunity to actually practice what we preach here with stem cell therapy on my knees. I was diagnosed with end-stage osteoarthritis about 3 years ago after a ski accident. And when I came into here to the Michigan Center about a year ago last month, the MRI was, I think it was maybe down to less than 25% cartilage remaining in both knees. So it was basically bone on bone. And I hadn't been able to run for a couple of years. And I've always been, having been in the Army and all the wear and tear that puts on the body, and then sports, before and after that, just a lifetime being pretty active. My knees were pretty beat up. So I came in here for stem cell injections last year. We did, I think, the forward edge of what the FDA allows in the U.S., at least up until very recently, which is the same day bone marrow aspiration and then autologous reinsertion of those stem cells into both knees. That was February of last year. And I went from, I think when I walked in here last year, I couldn't really walk downstairs without wincing a little bit. And I certainly wasn't running. And it took, after the procedure, it was significantly less invasive than a knee replacement would have been, which was the option that my hospital in New York City had offered me. Thankfully, I knew Dr. Navity here and the Michigan Center. So, and just about our work with Forever Labs, I knew that there was a better option. So I was stiff and sore for a couple of days. It was outpatient surgery. I think we did the bone marrow aspiration in the morning, the procedure in the afternoon, and I slept in a hotel that night. And then it took about, I would say, 4 months to really feel any difference. And it was just so gradual. Like, it's not like a switch flips, but it was just one day I realized, wait, normally what I'm doing right now really makes me sore quickly. And, I've walked like 3 miles and my knees aren't hurting. And then 9 months after the procedure, I ran a 5-mile trail run with no knee pain, which I couldn't believe it. Didn't feel sore at all afterwards. So it's been a year now, and I just had an MRI done about 10 days ago, just for a 1-year follow-up. And we don't have to go into the weeds on that now, but I understand that the results look pretty good.
Significantly improved.
Yeah. Yep. So very fortunate that I was able to benefit from that procedure. And that's what we want to support with Forever Labs. Just to be able to show that these emerging regenerative treatments are becoming more mainstream and, across America. And we're getting to the point now where, fortunately, people don't have to go to places like Panama or Costa Rica or throughout Latin America for these procedures. Now they can, thanks to legislation that's passed in states like Florida, they can start to receive those treatments here at home.
Yeah, and I think that's a great segue into why we're talking tonight, what we're talking about, which always driven us at Michigan Center Regenerative Medicine is using technologies that can drive to the best possible outcome.
Right.
And one of those variables is what dose we're able to inject patients with. Oftentimes now it's a limiting factor in how many areas we can treat in one day, for example. We won't know our dose of stem cells until after we pull your stem cells, process them, and then that day we have to decide how to distribute them. What if we could send those cells out to a lab and grow them just like you can any cellular material in a lab for weeks to months, and then have a significantly increased number of cells to work with? Well, now, not only we would imagine our outcomes go up for the area that we're treating, We can probably also add on additional areas on a single day because we have more cells. It's just kind of that simple.
Yeah, I think that's our limitation right now is, so we do have a limitation on how many cells we can actually pull, the volume. We're a little concerned about what does it do to the patient. So when we concentrate down the sample, we have as highly concentrated sample as possible to treat the area we're focusing on. But as most people know, when you get to a certain age group, you usually just don't have one joint. It's both knees. Sometimes it's both shoulders, knees, spine, etc. So, and when you go through the procedure, even though it's relatively simple, it's still a recovery process, right? So, and currently when we do a bone marrow aspiration, we have to wait a few months before we can sort of re-tap the bone marrow again, so it gets replenished. So with this potential technology, and some of it has to do with the regulatory process, the government that limits us with what we can actually do with your own cells. And going outside the country is always a concern. Part of why we do what we do is that we're trying to use somebody's own cells, their autologous treatment versus allogenic. There is always a concern. People are worried if they're older age, are my cells still good? And to be honest with you, yes, your cells are still good. If you can do it younger, obviously is better, but even somebody older, their cells are still good. Now, if we take those cells and we're able to expand those cells and utilize more of them, obviously, most of the time, more is better for what we do, but the higher concentration is key. We don't want to lose the quality of what we're doing either. So sometimes when we get some patients, they want to have several areas done at once. I always warn them that sometimes you're maybe not going to get the best result.
Yep. Oh.
Sorry about that. So this technology that we're looking at and the regulatory process is the biggest limitation we have right now. So when you go to Mexico or Panama or wherever, you don't always necessarily know where those cells are coming from. It's coming from somebody else, not your cells. So as Tom always says, that, they're testing for what we know about right now, hepatitis, HIV. But what about viruses we don't even know about yet, or tests we haven't come up with yet? Or are they really going to even test for these spike proteins, et cetera? So I think that would be a concern, especially going outside the country and having these treatments done. So we want to try to maintain our quality and safety here in the United States while using your own cells.
Can you guys expand a little bit on the two types of stem cell classes, autologous versus allogenic?
So yeah,
pros and cons.
Autologous means that you're using your own cells. So we take your cells from your body, and we either use those to re-inject, or with Forever Labs, we can send it down to the lab and take those cells that are yours, expand them, make more of them, versus the allogenic, which is meaning outside of your own body. So who knows if it's a placental or embryonic cells, or essentially not from you. So there is always a potential that you can have some type of reaction or some type of transference from someone else when you get those cells. So that's the big difference.
Can I ask a question about, we talk about the with culture expansion, the benefit is that you get to administer a larger dose of somebody's own cells because you're multiplying those cells in the lab. Like with the typical just autologous procedure where you're because right now the current law in most states allows that you take those mesenchymal stem cells out of a patient's bone marrow and immediately re-inject those into the patient. In my case, into the knees, but it could be any other location. But on average, I know everybody's different, but what's the expected volume of stem cells you might extract from somebody's bone marrow? And then with culture expansion, what's the optimal dose that you'd like to be able to inject for, say, for osteoarthritis or a herniated disc?
So when we take the cells from your own body, what we do is we count several different factors. So in the way the machine counts it now is by size. So when it sort of takes a view of the sample, it looks at white blood cells, red blood cells, platelets, and then it breaks down all the white blood cells into the subcategories of the white blood cells. We are thinking at this point that we're looking at the overall white blood cell count. So we like to be in the neighborhood of about 1.5 or higher billion, 1.5 billion cells, white blood cells. When we break down the categories of those cells, we're looking at specifically something called a monocyte. So we hope to get at least 100 million to 200 million of those cells. But when we're starting to do some very basic analysis, Tom's staff, he has some staff working on some research, we're actually noticing that it looks like the monocytes may not necessarily matter as much as the overall count of the stem cells. So we're trying to shoot for at least, so we go off of the white blood cells, 1.5 billion cells. And some of the subcategories are critical, like how many platelets are there, some of the monocytes. We're looking to meet some criterion as far as like, what is our minimum count for those things?
Right.
But right now we're thinking that the overall white blood cell count seems to be the most critical piece of it. That's for the stem cell procedure.
Mm-hmm.
There's a different, for the platelets, we're looking at a different categories, just platelets overall. And that's a different discussion. But again, we're looking at sort of a minimal amount. Now, there's some data that shows certain areas will respond better if they have at least the minimum number count of XYZ billion count. But right now, when we do our stem cell procedure, we're looking at the white blood cell count.
Understood.
Kevin, potentially for culturally expanded cells, how many cells can you guys culturally expand at a time? Do you know?
I think it really it's only limited by what the physician wants. To be honest, I think is the limiting factor is just time. So if we need to expand the number of available cells from a bone marrow aspiration or from that sample that we currently have in cryo storage, right? If we need to multiply that by 5x, right, versus 10x, it's just going to take more time. But there's really, in theory, there's really no upside cap or constraint on how many cells we can expand. I know there's something called, I believe it's called the Hayflick factor, which is the number of times that a cell can replicate before its ability to do that expires and it becomes senescent. I think for mesenchymal stem cells, I want to say, isn't that like in the 40 to 50 range, somewhere in there? So I don't see any scenario in which we would ever hit that. And I think once what the real opportunity for culture expansion is that it enables the patient to have access to these regenerative treatments for the rest of their life, because we've got that sample of their mesenchymal stem cells stored now, and we can multiply those basically on demand throughout the course of their life. Whereas now, under the current regulatory regime, it's basically it's one bone marrow aspiration for every procedure. So I had to do a bone marrow aspiration the morning of my procedure. If I wanted to do another procedure right now on my knees in any other state other than Florida, I'd have to have my stem cells pulled again. But with culture expansion, I think the real benefit is just the patient goes through one procedure one time, and then those cells are always available to be multiplied in the lab.
Yeah, I think that's a great point. That's why I was kind of looking from okay, what does this all mean? What does it do when you're in the exam room and you're talking to a patient and consulting them about their options? I think this is an important one to bring up. Like oftentimes your initial procedure is not your only procedure you're ever going to have in your entire life. Orthopedic conditions tend to accumulate with age. There are lots of other age-related diseases that stem cells are being studied for, may be available for. If we're doing a procedure today, why wouldn't we take some extra, put them in the bank, so that when and if you ever needed another one, they're already on ice, so to speak. They're already cryogenically frozen. We just have to unthaw them, grow them in culture to whatever dose we find that we need. And over time, we'll figure out which doses those are. And when you start counting stem cells, what is a stem cell? What's a total nucleated cell? I mean, we just need to start having the equation we use, or the nomenclature we use, needs to be consistent so that we can compare apples to apples. But the concept is you're getting more cells. And the theory is that should drive a better outcome. and it shouldn't lead to any side effects. We just haven't seen an upper limit where people start to get negative things happening from injecting too many cells. Worst case scenario, you paid for culture expanding a couple extra weeks, a couple extra months that you didn't need to do. But I think the safety is extremely well established. And now it's just a matter of how much better can we drive our efficacy and outcomes.
I think the key is, as far as safety is concerned, is it's autologous, right? It's your own cells. So there was a study that was done, I don't know, 8 or 9 years ago now, where they looked at allogenic cells, and they were injecting into the discs of a rat, and they thought, when we all think, it's America, right? More is better. So they did a disc with 1 billion cells, they did a disc with 3 billion, and they did a disc with 5 billion. They actually found that more was not better, and people that were getting the rats that were getting the higher concentrated cells actually were not doing as well. So now, again, these are allogenic cells that were in rats. Those cells were actually destroying the discs of those. That could be because the body only has so much it can handle. But again, this was an outside source cell, so who knows? And that's part of what we're trying to decipher is where's the sweet spot? But we're not even there yet, right? We don't even have the ability to make those cells right now. But if we could, it doesn't always mean more is better. It just means we can do more areas, right? And I think that's the key. And people, like you said, with orthopedic degenerative conditions, in my experience anyway, usually people just don't have a bad knee. They have a bad knee, they have a bad shoulder, they have a bad back, and there's some genetic component to these degenerative conditions. So, and how many patients I see all the time that have had their hip done, their knee done, their shoulder done, their back done, where as surgery we're talking about. So how is that different than what we do, right? Versus taking the risk. I mean, a lot of times these outcomes are pretty good, but unfortunately I had a patient that, she had one hip done. She went to have the other hip done, and this one went well, this one went terribly poorly. And unfortunately she's not doing as well because of that. So every time you go into the knife, there's always a risk, where when our procedures are, the risk is there, but it's not as great. The recovery is not as great. And in the end, there's still an option versus no option after surgery. So.
Yep. I think that's an important thing that we do in talking with our patients and looking at outcomes. We do track outcomes. We're getting great outcomes. Yeah. We're also tracking for adverse events, and we've yet to make anybody worse. We've yet to hurt anyone to the point where, they couldn't go on and have their surgery. And that's something operations just can't say. Even the best surgeon has negative outcomes. That's just part of the risk of surgery.
Can you guys talk about the current regulatory standards now for stem cells, using allogenic stem cells versus autologous, Florida versus the other states?
Yeah, I could talk about what we do with Forever Labs, and I mean the doctors here can chime in. So at Forever Labs, we only deal with autologous stem cells, those cells that are extracted from that patient. We just think the risks are too high with allogenic procedures for all the reasons that the doctors here have mentioned. We just focus only on autologous. The regulatory environment in America up until very recently has been, I think, under what's called like the Public Safety Act, was it Act 361, where the FDA regulates what are called human cell and tissue products, right? And they have, in order to do anything beyond what the FDA concerns or deems to be, quote, minimal manipulation, you have to have special approval. And so the procedure that I did last year, which is where my bone marrow is extracted, those stem cells are separated. No additives are included in the subsequent injection into my knees. We're just taking tissue out of one part of my body and immediately that same day injecting it into another. That same day autologous transfer. The FDA doesn't consider that to be any different than a skin graft, right? So that's the forward edge of what's been allowed. So culture expansion, you can make an argument that that's not really manipulating the tissue. You're just making more of those cells, but that's still been restricted by the FDA without special approval. So now what's happened in the state of Florida last summer, Governor DeSantis signed a bill into law that basically fast tracks stem cell therapy in Florida, where any stem cell drug or therapy that successfully passed a phase 1 clinical trial can now be offered in the state, subject to, there's a list of requirements. Obviously, very prominent disclosures to the patient have to be made. The cells have to be prepared in what's called a CGMP, or a current good manufacturing practices certified lab. You have the lab has to be certified by an independent entity, and you have to be registered with the FDA. You don't have to apply for approval, but you have to notify the FDA that you're doing this. So Forever Labs obviously checks all those boxes. We're currently building a lab in Florida right now that's going to be able to offer culture expansion in America. I mean, for all intents and purposes, for the first time, like at scale. And the opportunity here is to begin reshoring what's become, I think, the unintended consequence of these FDA restrictions has been the creation of this enormous market for offshore medical tourism, where anybody with the means is currently going to Latin America clinics in, say, Panama, Mexico, other locations, and paying a lot of money to do these procedures outside the United States. And the opportunity is now to be able to do this at home with all the protections of the American legal system.
Now, as far as I understand, allogenic is not necessarily approved.
No. Florida's only autologous.
Yeah, in general, I don't really know the regulation process, but from what I understood, it's not necessarily FDA approved, but I don't know enough about it, but people are using it, that being said. But from the last conference I went to, I still understood that allogenic products are not necessarily approved, but I don't know. Do you know differently, Tom?
I still think if you're doing and using allogenic products in the United States, it's not something I've heard of clinics getting shut down over. But I think if you were to have a negative outcome, it would be very difficult to defend.
Yep. Now, for current clients of Forever Labs that's banked with you, how soon are they able to use their bank cells for expansion?
Well, our lab's under construction right now, and we're currently on track to begin offering access to these therapies later this year. So I think the guidance we've received from our legal counsel is that both the culture expansion and the procedure need to be conducted in the state of Florida. So, for anybody around the country, if your doctor's willing to get on a plane, and if they have a Florida, or if they have a license to practice medicine in the state of Florida, then there's probably an option available for you. And it remains to be seen whether or not this will expand to other states. There are a couple of other states that have already passed similar legislation: Wyoming, Montana, and New Hampshire. I think Montana and New Hampshire have already passed the legislation. I think Wyoming's is working its way through the legislature right now at the state level. Remains to be seen whether or not this will be rolled out at scale in other states or even nationwide. We're paying very close attention, and anybody can go to our website to stay up to speed on what's happening on the regulatory front. We post regular updates whenever news breaks.
Excellent.
Yeah, I think we're doing probably pretty good on time here. I think we covered most of our topics we were hoping to cover tonight. Kind of in summary, this is a big moment in regenerative medicine. There's been a couple big moments since we've kind of been doing this. But this one feels like it's really going to change the needle, move the needle. Our injection techniques have changed. How we were processing same days have changed. All of a sudden, we're going to have a lot more cells to work with. And it's just, if you think about like an artist, like we got more paint, like to put on the canvas now. Like our pictures are going to get even more vivid. One of the, I would say, negatives that people, when I talk about regenerative medicine, is oftentimes our post-procedure imaging doesn't look all that different than our pre. people feel better. They're functioning better. We've definitely stabilized that degenerative curve. And I think that's where a lot of the pain relief comes from. And then people's function can go up because their pain's better because things stabilized. Maybe got a little bit better. You get a post-op MRI or 6-month MRI, and they look a little better. I'm hypothesizing those post-procedure MRIs are going to look significantly better once we find that sweet spot. Once we find that appropriate dose that joint needs, in addition to all the different techniques that we keep evolving, as our techniques get better, as our dosing get better, our outcomes are going to improve, and I think we're going to get more post-op MRIs that look like yours. You had possibly one of the best reactions based on an MRI of a patient I've treated. Yeah, it's incredible. We got to make a poster out of it. I've got to put it on the list of posters to create. But I'm excited. I'm beyond excited for this phase of what I think culture expansion is going to do for our field.
No, I will say something about the imaging. So I was just a little caveat, but sometimes it doesn't necessarily matter if it looks better. I mean, we want it to look better. I think the key is how people are functioning and how you feel. I mean, there's been dozens of studies over the past 20 years that have demonstrated there doesn't seem to be a correlation between what it looks like on pictures and what it feels like. And most of the imaging studies have been done in the spine, which, I've personally experienced, like looking at images and saying, this person, how are you walking, versus one that looks relatively benign and they're miserable. So patients ask me that too, am I going to see a difference in imaging? And I usually say, does it really matter if it looks different versus you feel better? I mean, it puts some validation to what we're doing. But I think the key is the function more than what it looks like, but it would make a nice poster, right? And so, but I think the most important thing is the overall function and how people feel, and what it looks like is just a bonus, I think. Mm-hmm. So, and I think it matters too. The imaging probably would matter more if we get to people at a younger age, right? If they're healthier and they're doing all the right stuff, their diet, their exercise program, that probably makes a big difference too. So I think it's multifactorial.
Mm-hmm. We have time for questions. Are there questions that came in, or we kind of running out of time here? We got our moderator tonight.
Well, you guys kind of covered a lot of the questions on here. Most of the questions are regarding allogenic versus autologous that we covered. So far, one of the questions is regarding insurance coverage, and we know that insurance still does not cover this yet. We're hoping that in the future, as more data comes out, it'll be one of those things that's going to be covered by the procedures, by insurances. But you guys kind of covered everything else. Kevin, thanks for your time, for coming down here. Really appreciate you coming down, sharing your experience, sharing us about the technology from Forever Labs. I think a lot of people need to learn about and hear about this technology. And hopefully a lot of patients come through, and this is the first time they're hearing about these treatments that's already available there. So thanks for coming out and sharing your experience. All right.
Thanks for having me, gents. Let's do it again soon.
ForeverLabs.com, Michigan Center of Regenerative Medicine, Regenerative Medicine Michigan.com. Any questions, send us emails, give us a call. We'd be happy to set up separate visits for anyone with questions or thoughts. And if they're interested in maybe becoming a patient, we'd love to take a look.